In this concept, we'll be talking about the nature of sunlight, photosynthetic pigments, the excitation of chlorophyll by light, photosystems, linear electron flow, cyclic electron flow, and chemiosmosis in chloroplasts and mitochondria. The sun gives off a lot of electromagnetic radiation. The part that humans can see is called visible light. It has a wavelength from 380 nanometers to about 750 nanometers. Purple is in the 380 range, red is in the longer 750 nanometer range. Blah, blah, blah. What's all this about with wavelength anyway? And what does it have to do with photosynthesis? Well, everything. For this class, I want you to know that the shorter the wavelength, the more energetic the electromagnetic radiation. The more energetic the electromagnetic radiation, the more energy it can pass on. Now, there is such a thing as too much energy. Electromagnetic energy in the gamma through most of the UV range have enough energy that they can damage cells, killing them or causing cancer in multicellular organisms. The electromagnetic radiation in the microwave and longer range have too little energy for cells to use. Visible light, as Goldilocks would say, is in the just right range for most life on Earth to effectively use for photosynthesis. So, visible light bathes the Earth, shining on photosynthetic autotrophs as represented by this leaf here. The leaf, as we see in cross-section, has pores or stomata for allowing access to gases like CO2 and oxygen. Cells of the mesophyll are packed full of an organ called the chloroplast. The chloroplasts have a double membrane. See, right here. Inside the double membrane are stacks of membranes. These individual folds are called thylakoids, and the space outside of them is called the stroma. Yes, all of these terms are important, so make sure you know them. Embedded in the membrane of the thylakoids is a very special molecule, chlorophyll. I won't make you learn the different types of chlorophyll, although we do cover the major groups in lab. I want you to notice three things here. Starting at the bottom, the hydrocarbon tail. Why would it need a hydrocarbon tail? That's right, hydrocarbons are hydrophobic. The lipid membrane of the thylakoid is hydrophobic. The hydrocarbon tail, or hydrophobic tail, allows chlorophyll to be embedded in the thylakoid membrane so it doesn't move around. Next up, the porphyrin ring holds the magnesium here. The magnesium is very important. Here's why. Energy comes from the sun and strikes the chlorophyll molecule. An electron from the magnesium gains energy and jumps to a higher orbital. We call this the excited state of the electron, or that the electron is excited. Here's an analogy that may be made. You have a 3-year-old that is wakened from a nap too early. He's cranky and has low energy. You give him a candy bar to try to cheer him up. Well, it cheers him up all right. He's bouncing all around the room in an excited state. The kid has received more energy from the candy than he had before. The electron of the magnesium has more energy than it had before because it has received energy from the sun. Well, now the electron has two options, because the energy from the sun is like the energy from the candy bar. Neither can last. The electron can go back to its original orbital or ground state and give back the light that it absorbed, minus energy lost through heat. We call this fluorescence. All glow-in-the-dark toys operate on this principle. The other option is for it to go on a marvelous journey through the chloroplast, donating the energy that it gave along the way. But before we talk about the journey, let's talk about where the electron will be starting its travel. Chlorophyll is found in what we call photosystems. It is a complex of pigment molecules, including chlorophyll, structural proteins, and electron-accepting complexes. Surrounding the chlorophyll are pigment molecules that harvest light molecules. This allows the photosystem to gather more light by having more molecules than just the chlorophyll at the center of the photosystem gathering the light. The pigments funnel the light they harvest into the chlorophyll. Here's another way to think of it. Antennas are used to pick up radio signals out of the air. A big whip antenna gathers more radio signals than the little antenna on your cell phone. That's why trucks with whip antenna don't need radio towers to relay the signal like your cell phone does. Back to the photosystem. The light is funneled or relayed to the chlorophyll, and the electron in the magnesium is excited. It is removed from the magnesium by an electron acceptor before it can fall back to its ground state within magnesium. From there, the electron is passed to another electron acceptor embedded in the membrane of the thylakoid, then another, and then another. At each stop, it gives up a little bit of its kinetic energy to do the work of pumping protons or hydrogen ions into the thylakoid space. Is this sounding familiar? It should. It's the same system that mitochondria use. Mitochondria get their energy-full electrons from redox reactions, whereas chloroplasts get their energy-full electrons from sunlight striking chlorophyll. Just like in mitochondria, the buildup protons are allowed out into the stroma down their concentration gradient through ATP synthase. This generates ATP the plant needs for the Calvin cycle, which we will be talking about in the next concept. Once the electron reaches the end of the electron transport chain associated with photosystem 2, it was discovered second, the electron is passed to photosystem 1, where it gets another shot of energy from light. Then it is passed through a second electron transport chain. Finally, worn out, it is passed to NADP plus reductase, which slaps it onto NAD plus to make NADPH. No, I haven't misspoken, nor is NADP plus in the figure misspelled. Chloroplasts use NADPH, mitochondria use NADH. One way for me to remember this is P for plant. It really doesn't mean that, but it will help you remember. P for plant, so NADPH in chloroplasts. The NADPH goes on to the Calvin cycle. So this goes on and on and on and on, with magnesium donating more and more electrons, right? Nope. Once magnesium has lost an electron, it now has a positive charge. It holds on to its remaining electrons until it gets another one to replace the one that it has lost. But where does it get it? From water. Water is split, the oxygen ripped from the hydrogens. The electrons are stolen from the hydrogens and given to the magnesium. The hydrogen ions contribute to the proton gradient within the thylakoid space. The lonely oxygen, which wants to share an electron, hooks up with another oxygen, forming molecular or gaseous oxygen. This is where we get the oxygen in our atmosphere. This is why this form of photosynthesis is called oxygenic or oxygen producing. That was the linear flow of electrons, or the flow of electrons one way through photosystem 2 to NADP plus reductase. Sometimes the plant doesn't need NADPH for anything, but it needs ATP to run its anabolic reactions. Then it uses a cyclic electron flow. Light excites an electron in magnesium in the chlorophyll molecule in photosystem 1. The electron is removed to the primary electron acceptor, which uses the energy from the electron to pump protons into the thylakoid. The electron is eventually passed back to its original magnesium. The electron has a round trip rather than a one-way job relocation. The round trip is called cyclic electron flow. Before we begin this concept, let's do a quick summary of chemiosmosis. Chloroplasts and mitochondria both use electron transport chains to create a proton gradient across a membrane. The protons, being charged, cannot get past the membrane, but they want to go down their concentration gradient to the area with fewer protons outside of the membrane. The enzyme ATP synthase, which is found in both chloroplasts and mitochondria, allow the protons to move down the concentration gradient. The energy from this movement generates energy to regenerate ATP from NAD and inorganic phosphate. This process is called chemiosmosis in both chloroplasts and mitochondria.